All right, so let's do some of these, and you could laugh on your own to look at these things over here, molecules you might not be aware of, all right, that kind of thing. But you can look at it later on. All right, so we have to deal with chemical reactions. Now, also too, you're going to also see certain things the way I'm doing this course. There's certain things that are out of order, but I guarantee I'll be talking about it by the end of the course. One example is, in here we're going to talk a little bit about the DNA molecule, but I'm not going into the detail about it, about where C goes with G. If you've done that before, I'm not doing that here. I have a whole chapter. I'd rather spend more time when we talk about DNA and DNA replication. It'll make more sense there. You'll know a little bit about the DNA molecule and what it is, but I'd rather spend more time talking about, so I'm extracting some of this stuff from this chapter and putting it in another chapter. You'll still get it. It's just, I think it works for me better the way I teach. You'll eventually get it, okay? Also, too, I think enzymes were taught in another chapter. I think it was like chapter 1 or 2 or something like that. I left it blank, but I put it in here because an enzyme is a protein, and we're going to talk about proteins and reactions in this chapter. So I just think it's all going to be in the same text. Well, not the DNA thing, but you'll still get it by the end. So I might have done that. Does that make sense? It's just, for me, the way I teach, it's a little, I think it'll run smoother the way I teach. All right, so chemical reactions. We talked about this before. We're going to expand a little bit more. It's a process that forms or breaks these ionic or covalent bonds, right? And we talked about the symbol, how we symbolize it with reactants and products, where this is an irreversible reaction, but when we have the yield sign going both ways, then it's reversible. And I showed you a few examples of that. A good one is that carbonic anhydrous reaction that you've been seeing so much of, right? A little tattoo that I'm not encouraging anyone to get. And we have classes now of reactions, and we talked about these briefly, where we could have decomposition, large to small. We could have synthesis from small structures, and we get a larger structure. Or we could just do some kind of rearrangement and exchanging of things and creating new subjects over here, new products over here. But we now have to expand a little bit more with this. Energy in it, does water go in, does water go out to do this, that kind of thing. So we got to spend more time on that. So this is decomposition, synthesis, and exchange, and we'll see where this goes in. So we have metabolism, which is all the chemical reactions in the body, and we have anabolism. Anabolism is like anabolic steroids, right? Big, right? So these are going to be from small products getting into big products. Okay? Simpler to complex. This is anabolism, or an anabolic reaction. Okay? So it's a synthesis reaction. And in order for this to happen, water has to be removed, and I'll show you pictures. Water has to be removed, like an OH from one structure and an H from another. That makes water, that comes out, but it leaves a bond that needs to be made and they fuse together. That's why we call it dehydration, because we're removing water to do that. Okay? And energy is added to do this. Okay? Catabolism, think of catastrophe, right? Breakdown. So we're going from a big structure to smaller structures. We call it a decomposition reaction. And in order to do this, we have to put water into the reaction. And what's going to happen is that water molecule is going to break up. You have this big structure, whatever it is, and you want that big structure to break up. So there's water going to come in, and OH from the water is going to go someplace in the middle of that, break that bond, and then the H from the water is going to attach to the other one. So we're going to break up that water, it has to be put into there, to make two smaller things. So we call this hydrolysis. Hydro meaning water, Lysis means to break up. So we're going to take the water that we added, break it up, and fuse the parts to the two structures, and now you got two. So water is usually needed for this, and energy is released to do this. So let's look at this. Okay? We have up here some kind of chain. Anything that's a chain is called a polymer. Okay? We'll get that later. So you have this short polymer chain, and then over here we have, and think of this like, we could have like carbons here. Think of each one of these balls as carbons. And then this is a carbon over here. Well, we want to attach this monomer, which is just one carbon structure, and attach it to here. So you have two smaller pieces to make one larger. Well, we're going to take a hydroxide functional unit off of this, right? Alcohol. We're going to take that off of this and a hydrogen off of this. We put that together, that becomes water, and then we can have now this fused with this to make a longer polymer. Does that make sense? So water gets released, dehydration, for a synthesis reaction. Is that clear? Okay? Let's do the opposite. Here you have this long polymer, which was this. Okay? And over here we have the hydroxyl end over here. Well, what we can do is if we add water, the water can be broken off into OH and H. The H can go onto one part and the OH can go to the other part over here. We have to add water if we're going to decompose, to break up. And the water molecule has to be broken into two pieces. Hydrolysis. Hydro means water, lysis to break up. Is that clear? All right. So that's a little bit about these reactions. Now, it's the same concept, but now instead of looking at balls and sticks, we're now looking at now you're looking at actual molecular structures. But it's the same thing. Anabolism is this, where you're going to take two pieces over here, water is going to come out, and you're going to fuse it here. Catabolism, not catabolism, catabolism. Oh, okay. Catabolism is where we're going to go from one structure and break it up into two, but water has to be added, but you're going to break up the water and do that. Same thing. Same concept. Bigger structures, complex structures, same thing. Here you have two rings. If we could get a water here, OH and an H, water comes out, dehydration, and it fuses here. You want to break it up, add the water. You got catabolism, and I'll break it up over here. Same thing. Again, I'm not going to actually draw structures, but you should be able to recognize structures and how things work. Okay? All right. So now you know about reactions. So how can you speed them up? that can speed up reactions. You increase the temperature. You put an egg on the skillet or the pan. You put heat to it. It starts solidifying. Putting heat to something is going to make a reaction happen faster as opposed to letting the egg sit there for 2 or 3 days. That's not an experiment to do. But you put heat to something, it will go faster. You decrease the particle size. That will also increase the reaction rate. Now, let's talk about surface area, which has nothing to do with surface tension. Hint, hint, don't get mixed up with the two. But let me explain to you what surface area is. I would say 80% of students have problems with what that is. They just try to memorize it, but they don't understand what it is. This is surface area. You take it, let's see, should we do ice or should I got so many in my head. All right, let's do the potato. You take a potato and you dip it in oil. Now the only place that that oil can go, and you're cooking it, the only place the oil can go is on what we call the surface area. Okay? Can't get inside really, it's just on the outside. So it has a certain surface area. Now, let's take that same potato and cut it up into chunks. Okay? If I take this structure, okay, the only place things can hit is the surface area. So there's a certain surface area here, right? Our potato. Now I cut it in half. I created 2 new areas that things can touch, right? Can you see that? I increased my surface area. I didn't change the volume of the structure. I increased the surface area. So now I can touch that area. I created 2 new surface areas. 2 more. Do you see how the surface area is increasing? By just breaking things up. Let's go back to potato. You have this potato. You put it in hot oil. The surface area is going all over the potato. That's the surface area that oil is going on. Now I take the same potato, I cut it up into chunks. Surface area is going up now, right? There's going to be more oil going onto the potato because there's more surface area. What about if I take that same potato and I slice it into very thin sheets? What's the surface area like now? Because I can hit on either side of all these little slices. Is that more than the chunks? You bet. So there's going to be a lot more oil on these slices. And this is why potato chips are worse off than there is French fries, is worse off if you take a baked potato and you actually put that with oil. Because there's less oil on it. You see that? Does that make sense? You understand what surface area is? Let me do one that's actually quicker to understand too. You take a block of ice and put it on the stove. The only area that the heat can really get to is the surface area. Okay, whether it's outside in the kitchen or on the stove, the only area, kick it inside. That's the surface area. But now I take that same block of ice and I turn it into ice chips. And I put that on the stove. Which one's going to melt faster? The ice chips or the block of ice? You do this all the time, you just don't know what happens molecularly. It's the same thing. So if you decrease the particle size, you're increasing the surface area, which means that you're going to increase the reaction rate. You just prove it to me when you did it with the ice block and the ice chips. Be clear with that, right? Are those good examples? Who had no idea, or like you thought you had the idea what surface area is coming in here, but now it's like, well, now I know it now, right? That's rhetorical, right? It makes a big difference. Okay. All right. What other things? If you increase the concentration of a certain substance, the reaction is going to go faster, right? If you put a little salt in the water, it'll boil faster. I don't want to explain how that happens. If you put more salt in there, it'll move faster. So if you increase the concentration, that'll also increase the reaction rate. So all these things will do that, okay? We also have things called catalysts. We can add things to things for the reaction to occur, or occur faster. So if you increase the reaction rate without being, and this could be added to the reaction, and yet this catalyst doesn't get disturbed. It's like that salt that I just said. If you put salt, like you just let the water boil, it'll boil eventually. You put a little salt in there, it'll go faster until it all evaporates, but the salt will still be left there and you can reuse the salt. Does that make sense? That's what a catalyst is. So why am I talking about this? Because we have biologic catalysts. And we've been mentioning that before, but here we're starting to define things. Enzymes. Enzymes are proteins in our body, and we have, what's the last count? I don't even know. Thousands. Thousands of enzymes. We mentioned the one before with the carbonic anhydrase reaction. That reaction, by the way, will happen by itself, but it would take days for that to happen. But if there is something called carbonic anhydrase, which is an enzyme, it happens in a split second. Little tip for you, if it ends with ASE, it's an enzyme. Amylase, kinase, maltase, da-da-da, keeps on going. Okay? So enzymes. So we got to talk about enzymes now. All right? They're proteins. And we'll explain what those are a little bit later. Proteins that function as these biological catalysts. They allow us to do reactions that happen very fast with our own body temperature. Okay? Whatever that normal is. You don't have to make your body temperature go high or low. So we have a substrate, which is the substance an enzyme actually attaches to. And it lowers, oh, we got another energy thing here called activation energy. It lowers this activation energy so that it can get the ball started, okay? Let me explain to you what this is. It's very easy to understand what activation energy is. If you got to go from the reactants to the products over here in a graph, well, you got to push it a little higher. This is the amount of energy. You have to put more energy to get it over this hump to get to the products. And that hump we call activation energy. You got to put a lot of energy just to get it over there. Well, what an enzyme is going to do is that you don't have to go so far up a hill to do that. In other words, think of what this is happening. And think of this as a hill. I have a car over here, and I got to get to this point here. I got to go up this hill. I got to put more energy stepping on the gas for a longer amount of time to get it all the way up there. And once I get up there, it goes down. Now, I could do that with an enzyme. It'll work. But it's a lot of energy. But what the enzyme is going to do is going to cut down on that hill. Drop it down so you don't have to put that much energy into it. And then it can go faster. And that's basically what enzymes do. It lowers the activation energy, less energy to put into this reaction, and the reaction happens astronomically fast. And in a the meantime, the enzyme can be reused. And it's untouched, really. So here's the enzyme action. Let's give you an example. Sucrose is table sugar. And it has two rings bound to each other. One ring is going to be glucose, another one's going to be fructose. But they're bound together, now it's known as sucrose, table sugar. And what's going to happen is, you can read this on your own, but let me just show you the picture, it just makes sense. This cloud here is the enzyme. And the enzyme of this reaction we're doing is called sucrase, right? If it ends with ASC, it's an enzyme. So sucrase is an enzyme to break up sucrose. If it ends with OSC, it's a sugar. There's little reasons to understand all this and make life easier. So look what's going to happen here. Here's sucrose, which is two rings. When it goes into this area of the enzyme, the active site, the enzyme does a reaction. It lowers the activation energy, and when this sucrose comes off, it comes off as two parts. Now, don't get me wrong, that sucrose can happen by itself to break up. It could take hours, but you'll be dead by the time that happens. You need to break up and digest food fast. You need enzymes because these happen in a matter of milliseconds. And that's how enzymes work. Okay? Again, what's nice about enzymes is that once it does its job, it goes off and finds another sucrose or whatever it needs to do and break it up. It doesn't get destroyed in the process. It's this reusability. And the enzymes are not consumed. They don't use the energy either. They're lowering the energy. And I told you, it's really fast, astonishing fast how these enzymes work. All right? A couple other things. Cofactors. About 2/3 of human enzymes require some non-protein cofactor. Inorganic things, things that don't include carbon, iron, copper, zinc, magnesium, calcium, the things that you eat, that you hear is good for you. Well, certain enzymes need those things. Some of these organic things, these cofactors, bind to the enzyme and induce a change in that enzyme so it becomes more activated to do its job. And we need these cofactors to live. We also have coenzymes, which is basically the same thing as these cofactors, except they're organic. They're going to be things that are going to aid in these enzymes to do their thing. And those are a lot of your vitamins, niacin, riboflavin, and stuff. Just to give you an example, this is the lecture I dread to teach. But just to give you an example, that this here is a coenzyme. It's going to carry an electron from a glucose to bring it over to another reaction to create something to keep us alive called ATP. You'll learn about that later. I'm just giving an example. That won't be on your test at this point. It's way over scope at this point. But I'm trying to show you things of how these cofactors would be used.